think all of the advances in genomics and genetic engineering that have happened over the last few years have really changed what's possible in thinking about biomedical science.
We have the sudden and exponential improvement in cost and quality in both reading and writing genomes, maybe 1,000,000-fold for each of them.
If we create synthetic creatures of some sort, are we going to know, and are they going to know what's good for them?
This is that genesis moment of an evolutionary shift, a step forward for the history of human beings.
Will there be a point where it's fundamentally the right to create life form? What kind of world is that that's going to improve that?
We have to be careful what we create, but that goes for product development of any sort.
For every decision we make, there are consequences.
Hello, Morgan.
Morgan isn't, to me, really a sci-fi movie. It's something else. It gets into more of a philosophical discussion. The one thing that really kept me going through it was trying to understand what human evolution was really going to look like. And Morgan, in a sense, touches upon some of that.
Evolution has done a very mixed job for us humans. Our genetic makeup creates faults. The cells mutate, and all of these things means that many people die of terrible diseases, many people die prematurely. I am hoping that we can improve on that, and one of the ways we might do that is actually by something that we have just created really in the last decades or so, the power to do, and that's intervene in the course of evolution.
There's always a tension between what's perceived as natural and science, and sometimes that just comes down to, I think, people's understanding. For sure, human beings have been genetically modifying organisms ever since we first chose our favorite piece of corn out of the ground or chose the favorite wild pig to cultivate. So the animals we have and the plants we have today would not exist apart from the selective breeding programs that humans have imposed on them. So we've been genetic engineers for as long as we've been humans.
Genetic engineering is a sort of blanket term used for a wide range of attempts to influence the genetic makeup of human beings. But I think what most people have in mind is when deliberate attempts are made to delete particular genes in our genetic makeup and substitute others, or simply delete ones that are harmful and hope that the result will be beneficial.
Inside all of us, inside our cells, we have these great big long pieces of DNA that contain genes. Genetic engineering is the idea of going in and tweaking those genes or making changes to somehow improve or change the characteristics of the organism.
The Human Genome Project was actually first launched back in the late 80s, early 90s. And the idea was to have a 15-year-long project that would uncover the DNA sequence that actually made us who we are. The idea was to try to use that to actually decode all 3.2 billion bases of human DNA.
Keep in mind, the International Human Genome Project, after 13 years, right, took billions of dollars to really get an aggregate of like one human genome, right? But now you can do that routinely in labs.
Since that time, we've actually had newer technologies that actually enable DNA sequencing at a much, much more rapid pace. In fact, today you can actually sequence an entire human genome in about an afternoon, and it costs less than $1,000.
In 2003, we got the first ever human genome, and now, you can take a bit of your DNA and send it to companies, and they'll analyze your genes and send you back information. So that's kind of cool, and it's fun. But there's something actually much more important that's going on there, that as we are starting to get genetic information from more and more people, we are now going to be able to understand the basis of different diseases that we didn't know before.
The human genome consists of just 4 chemical letters that are strung together to actually make up the entire sequence of human DNA. I think of it sort of like a programming language, which is the cell is like a computer. It interprets this code to actually give rise to a very, very complex function. That is, to encode all the diversity of all the cells in our body.
Cell surface, big ones. The genome is like a book, right? And one can imagine that it's a blueprint, and so you have to decode it. Sequence genome, sequence DNA, basically output a string of letters. ACGT, right? String of letters that gets read by computer programs. There are many algorithms that enable you to compare different genomes. The letters, the order of them, they can be compared. And we use large compute clusters or farms, numbers sometimes in hundreds to thousands of computers or more, that simultaneously crunch these genomes and make comparisons and try to make sense of all this genomic data.
We need to be able to understand how that information is processed by the cell, and we need to be able to use that information to actually write new programs that carry out new functions.
One of the really exciting things that's happened in the last few years is we've got this great new tool in gene engineering. It's called gene editing, and one of the examples of this is a type of enzyme called CRISPR-Cas9. I just think of them as genetic scissors. What this allows us to do is, with very high precision, go into a cell, go into all the DNA in the cell, and find just one gene that we are interested in, and then we can make changes to that specific gene.
You can now design in the computer 20 A, C, Gs, and Ts in a particular order to your specifications, and it will find that needle in a haystack in, say, a human genome of 6 billion A, C, Gs, and Ts. It will find the 20 that match, and it will cut and then substitute in exactly what you want. So that's real editing, is precise changing of 1 or 2 or many base pairs to whatever you want.
Gene editing gives us this unprecedented level of precision to make very specific changes that we haven't had before.
So gene editing can be used to change any DNA in our body, and it can be used to affect pretty much anything, from the color of your eyes to repairing genes that are associated with certain diseases.
We're already well on our way to moving beyond these small changes that you get with CRISPR to very large-scale genome engineering projects ranging from bacteria to human genomes. In this case, you can hop in, you can replace huge chunks up to millions of base pairs, and those can be completely synthetic or they can be semi-synthetic, completely designed in the computer. So rather than changing one base pair as you might typically with CRISPR, you can change millions at once.
My research is based on trying to genetically engineer a certain type of cell in our bodies. It's a stem cell that lives in our bone marrow, and these are the cells that produce all of the cells in our blood and immune system. And in particular, we are trying to engineer them so that those cells are now resistant to HIV. I can take cells out of a patient, I can genetically change them to fix the genetic defect they have, and then we can put them back into the patient. There's many diseases that humans have figured out how to treat and how to cure, but the big ones, they're still there. And I don't know that I think that drugs are ever going to get us to the stage of being able to treat complex diseases.
The neuronal markers are they're relatively consistent.
We have a focus in part of the lab on neurodegeneration. And so neurodegenerative diseases such as Alzheimer's disease, Lou Gehrig's disease, ALS, you have mutations in proteins that essentially cause the neurons to die over time. And so we model the disease by creating cells that have the mutation. So we use genome engineering, CRISPR technology to create the mutation that you think causes the disease.
I can see in the future that once we have that information, we will very quickly be able to develop a gene therapy that will go in and change the genes that are causing the problem and maybe make people who would otherwise get ALS not get that disease. That's what I think is really exciting, the potential to use gene therapy combined with the knowledge about the genetic basis of disease to really get out ahead of diseases and make them not happen in the first place.
For those of us who study diseases, I think this is a very exciting time. You can fix these different diseases, right? But there's also a debate. The same technology, right, can be used to make someone stronger, make someone's bone harder, increase the IQs of people, and live longer, age slower, right? So those are all mutations that eventually we could learn how to make. That becomes a very interesting debate, right? Whether we should.
We humans, I believe, are absolutely compulsive self-improvers. If we could design our children to be healthier, smarter, more durable, more resistant to disease, less vulnerable to climate change and all of these things, that might not only be necessary for our long-term survival, but it might make the lives of our children and ourselves better.
If you're blending that with improvement of DNA, improvement of medicine to fix the newborn medically before he even comes out the womb, the thing is that's rubbing shoulders with creation.
Morgan raises some interesting scenarios. I mean, can we get accelerated development, for example? And our lab does exactly that. We have some developmental processes that normally would take hundreds of days, and we can do it in 4 days. We can essentially skip over many intermediate steps. Now those are at the sort of the cellular level where we're making, clusters of brain networks.
The scientific downsides to thinking about enhancements is that, we still don't really understand how all of our different genes interplay. And what that means is that as we make mutations, for example, in certain genes, we may be thinking we're improving intelligence, we might be giving people superhuman strength, but we don't understand enough about how all of these genes interplay to know that we're not creating other problems down the line. And so I think that genetics isn't black and white. There are a lot of gray areas, things that are not quite bad or good, but that in different contexts have, positive and negative aspects.
We're at an amazing point in synthetic biology, which is really due to the exponential decrease in costs and improvement of quality of both reading and writing genomes. And what that turns into is a whole variety of new tools and applications.
We live now in an age where we can synthesize life, right? So Craig Venter has shown in 2010, right, that he can create organisms from scratch.
In the last couple of years, they've synthesized an entirely synthetic yeast. Now, yeast is a living organism, albeit it's a small kind of mushroom thing, but nonetheless, it's entirely artificial, which is, this is really playing God.
Maybe we could actually go all the way up through layers of complexity and make bigger organisms. Maybe we could even make humans.
There are actually efforts now underway by leading scientists that's called the Human Genome Project, RIGHT, which is attempts to actually synthesize the entire human genome, not just to read it like the Human Genome Project did, but now to make it.
Morgan.
When we contemplate creating a synthetic individual with the sort of levels of functioning that we have, along with which go the possession of rights and responsibilities. So the real issue is, if we were to create a synthetic or even partially synthetic creature that had powers and capacities like us, in the wonderful phrase of the great 17th century English philosopher John Locke, that had reason and reflection.
Do you consider yourself a person, Morgan?
No. I am something new. There is no appropriate label.
I think developing something like Morgan is evolving over time a stable emotional landscape, a sense of soul.
You experience love?
I have the ability to demonstrate love.
Do you have something missing if you're created in a lab? You have what we proudly talk about being a soul. Can that be implanted or evolve inside a creation who at the moment is super bland, very, very physically strong, powerful, can do the 100 meters in about 8 seconds, has no feelings. And the second I pop that tablet into its head and it's so in advance of me, you've created a time bomb. And therefore, the time bomb may be so smart as to not reveal its true feelings. So once it's not revealing its true feelings, you're in trouble.
What is that? Show me how you feel!
If we created a synthetic individual, then it seems to me that that creature would have just the same rights as other creatures, like humans. It would give us no rights to control them any more than we have any rights to create new human slaves.
Do you know the cruelest thing you can do to someone you've locked in a room? Press their face to the window.
I think we all have to have a say in what it means to be human, what it means when we have the capability to change things, and whether or not we should go down these paths. Sometimes just because you can do it doesn't mean you should. But also, I think it's important that we don't have a knee-jerk reaction and just say, whoa, this is playing God, we can't do it.
Is this what heaven's like? I don't know.
Because I think our definition of playing God changes every decade, depending on what our capabilities actually are.
What really excites me about the future of genomics is that now that we can actually sequence a human genome for less than $1,000 means that we can actually start to decode each and every one of our genomes. We can start to decode the genomes of different cancers, of different disease states, and actually understand how each of us differ and how to treat each individual person in a very unique, personalized way.
I see in the future that we will be able to come up with customized gene therapies in the same way that you can have a customized drug regimen that's prescribed for you that will just make sure that you don't develop diseases that your genetic potential says that you probably would.
I think there's going to be a great deal of pressure on biomedical research to produce therapies that prevent or reverse aging processes, including cognitive and physical decline.
I think genetic engineering has the potential to change every aspect of our life. Obviously, it's used, for example, in agriculture and has improved food production in many respects. It will impact biomedical research and treatment of disease in very concrete ways over the next few decades, in ways that are even hard to imagine.
Do you think it's safe to say that your consciousness is superior?
No, it's just different.
How we are impacting the evolutionary process, not just synthetic biology, but even more broadly than that. Evolution, which includes cultural and technological evolution, is going faster and faster.
We're looking at an idea that really asks the question, what is the next evolutionary state of a human being? And what will it look like? And what is the flaw? What is the flaw in that particular design? Which is a beautiful thing, because we're all flawed, and that's why we want to evolve.
I know why they made me.
A number of people are very worried about the creation of artificial intelligence, and particularly in creatures that look like us, that have preferences, the desire to make choices, to have ambitions, to change the world, which is an ambition that we recognize as legitimate for human individuals.
The big challenge, of course, is that biology is really complex, which means really approaching it with some amount of caution and understanding the sort of interplay between various genes and how that can actually give rise to unintended consequences is really important.
We have to be vigilant not to cut ourselves off from technologies that may assist us to survive, but also vigilant not to create technologies that will be the end of us, the death of us.
We can hope for the best and maybe spend some time planning for the worst just in case to avoid it. But I think that we're really entering a very exciting time.